Well, good afternoon. My apologies to start that I don't speak Spanish. I have trouble also with English, so I hope you'll bear with me. I want to thank the representatives from the government, from private sector, Neil Reardon, my old new friend, for inviting me. This is my first trip to Panama, and I'm hugely excited to be here and to contribute to this very important meeting. So this is my obligatory disclosure. I started a company called Osiris Therapeutics. I've been divorced from Osiris for a number of years. That means that I steal some of their data to talk about the science, but I'm not a consultant or a salesperson for Osiris, although there'll be a point where you think I'm selling their products. I also started a company called Cell Bank Technologies. I'll tell you a little bit about that, and I have no other conflicting areas. So I want to start with this slide because it's a very important slide. It plots the number of scientists as a function on the bottom scale of the R&D as a function of gross national product. And you can see this big, huge blue ball is the United States. We have a huge industry in the United States, and the government and private sector contributes huge amounts of money to this new technology. If you look in the lower left-hand corner, you can see the Americas, and Brazil in particular has a very small percentage of its gross national product committed to R&D. But I think that the important aspect is that every country, in order for it to develop its own innate technologies, has to make centralized investments. And I think what's going on in Panama is the right way to start. I think Panama needs to increase and steal some money from those ships that are coming through the canal to increase the technology base and to encourage the formation of high-tech jobs for its citizens. And it's a way in which a Central American company, a Central American country can actually come into its own and establish its own identity amongst the world's technology base. So I want to make two other important points. One is insulin has been used for over 100 years to treat diabetes. Insulin will not cure diabetes. Likewise, nitroglycerin has been used for angina since the 1880s. It will not cure or affect the defects that the heart pain are telling us. Stem cells and the stem cell industry, both in academia and private sector, have an opportunity actually to cure these diseases, and that's what we're going to talk about this afternoon. The FDA in the United States and the regulatory agencies in Europe regulate cell-based therapies as if they are drugs. This doesn't make good sense. You can take a mass spectrum of a drug and show that it's 100% pure. You can't take a mass spectrum of a cell and identify anything in that cell. So regulating an industry by the standards of drugs for this new emerging cell-based industry, these new biologics, for me, needs to be reevaluated. And so my colleagues and I, Mike West and Andy von Eschenbach, have proposed what we call progressive approval. In this case, you do a safety study, phase 1. You don't do phase 2, you don't do phase 3. The government and the regulatory agencies approve the product for sale so that small companies where real innovative technology exists, small companies can have a cash flow and can actually support what we would call a phase 4 post-marketing follow-up study where the regulatory agency would determine what parameters will be measured. And I think the innovative piece of this is that all of the patient data that's acquired be put on a publicly accessible website in real time protecting the identity of the patient, but all of the positive aspects of any technology can then be viewed when they reach statistical significance. It'll be obvious. And also when there are adverse events, guys like me off somewhere else looking at the internet can help find out what caused the adverse events. And in this case, the whole scientific and medical community can be involved in all of these clinical trials and all of this new technology. This is a proposal that will be made to the FDA, will be made to the European Parliament, and I think Panama is a place where this could take place today, and you could show a leadership position. So I'm lobbying with the appropriate governmental authorities to have an open mind about new ways to regulate technology, because the technology is becoming very complicated. So stem cells are in the mainstreams. Whenever you see some topic in Time magazine, you know you've made the big time. So there's this new subspecialty called regenerative medicine. Regenerative medicine comes in two flavors. The first flavor is called tissue engineering. You take cells, you put them in a biological matrix, and you surgically implant them to either cure a problem or generate a new tissue. I'm not going to talk very much about that. I'll talk a little bit about it. But what I want to talk about is this other kind of regenerative medicine. This regenerative medicine dates back to the Greeks, to the god Prometheus. Was a god who angered the other gods, and every day the other gods sent a bird of prey to eat one of his organs. Luckily, they chose liver, which has a very high innate regenerative potential. But let me tell you that all of you are regenerating body parts as we speak. So how do we control and organize the regenerative potential that you innately have to provide medical care? And this is a perfect example. Is every second 15 million blood cells in your body drop dead and are perfectly replaced without you even smiling. So while I said that sentence, a couple of 100 million cells drop dead and were perfectly replaced. That's your innate regenerative potential, and that's because on the left side of the slide, you see there's a cell in your bone marrow called a hematopoietic stem cell, and it gives rise to all of your circulating blood cells. If you lose your hematopoietic stem cell, you will die. You will drop dead because all of the other blood cells will die as is properly scheduled for those cells. So I've suggested that there's another stem cell in bone marrow, which I've called the mesenchymal stem cell. And this is a hypothesis slide that I fabricated in the late 1980s. You have to, most of the people are too young to remember that in medical school, when I went to medical school, we were taught that there is one stem cell in your entire body when you're born, and that's the hematopoietic stem cell. When your brain will have all the neurons you possibly can have, and because of all of the drinking of red wine, you're going to kill many of those neurons, and that's called aging. And it turns out the same was true for heart. So heart, when you're born, you have a certain number of cardiac myocytes, and they either get bigger or smaller, but they never change number. That's wrong. Even Neil Reardon has neural stem cells in his brain. Even Neil has stem cells in his heart. It's hard to believe that he has any stem cells in his heart, but they're there. And actually, they're slowly regenerating his tissue. So how do we control that innate regenerative potential? And that's what this slide is all about. It turns out if you want to do tissue engineering, you need to know about these lineage pathways that make cartilage or bone or muscle or tendon. So your ear cartilage, your nose cartilage, your throat cartilage, your sternal cartilage, your hip cartilage, your knee cartilage, your ankle cartilage, all different. You give me a slide, I can tell you which one of those cartilages you have. So how you come down this lineage pathway into those different phenotypes will determine your success or failure as a tissue engineer. For innate regenerative potential, the top of this slide is the important thing. It's the stem cell, the mesenchymal stem cell at the top, and what it can do, which is the most important parameter. And I'm gonna give you the end of the lecture now, so that for the older members of the audience, if you need to take a nap, wait 5 minutes, listen to the end of the story, because the end of the story will tell you actually how you have to think about these cells, because the story has changed over the years. So this is, you have to memorize this. All MSCs are pericytes. They sit on every single blood vessel in your body. When you look at all of these scanning EMs of different vessels, every one of them has pericytes on it, and depending on where that vessel is in your body, the pericytes take on different morphologies. They function in exactly the same way, and they function when there's injury. The pericyte comes off, and that pericyte then turns into an MSC. That MSC becomes activated. The activated MSC at that particular site secretes 2 classes of molecules. One class are immunoregulatory. They put a curtain in front of the cell. This is not systemic, this is local. They put a curtain, and immune surveying cells cannot get through that curtain. So it's your first line of defense against autoimmune reactions. It's what you set up, because when you injure a tissue, you liberate new antigens. And so these nosy T cells want to get at those new antigens, and the activated MSC puts up this curtain, and I'll show you why that's important for clinical use in a minute. Secondly, they produce from the backside molecules which I call trophic, which are anti-apoptotic. So you break a blood vessel, you have ischemia, cells die of ischemia. The MSC produces molecules, I'll show you what these molecules are, that stop the cell from apoptosing due to ischemia. They're anti-scarring. The cells that come in that produce huge amounts of scar tissue, the MSC puts out molecules that inhibit their entrance and inhibit their secretion of type 1 collagen. They're angiogenic. The MSCs make huge amounts of VEGF, but also these new fragile capillaries, the MSCs come and become pericytes again and stabilize those vessels, angiogenic. And they're mitotic for stem cells, as we'll hear. Cardiac stem cells are the MSCs cause them to divide in areas of injury and actually repopulate sites where other cells have died. So immunomodulatory, trophic, that's what MSCs do. And so since I invented this name, MSCs, of course my delicate ego, I want to keep, everybody needs to keep calling them MSCs, but what they do in your body has nothing whatsoever to do with stemness. These are medicinal signaling cells. These are drugstores for sites of injury. They function at these sites to do this immunomodulatory and trophic activity. So MSC, keep calling them MSCs, please. It's required for my ego, but they're not doing stem cell function when they're doing these activities. It has nothing to do with stem cells. I'll come back to that. So for someone who worked on marrow, I was appalled over the years that people were getting MSCs from fat, from heart, from skin, from brain. How disgusting from fat. I can't imagine anything worse than an MSC from fat. Why is that the case? Because they're pericytes, because they're on every blood vessel of the body. And therefore, it all makes sense that you should be able to get MSCs from any tissue that has blood vessels. So we can isolate pericytes, specifically sort them And here's a sort. The bottom, I know the videographer is not gonna like this, but tough break. If you look at this bottom line, the sort is for CD146, which is a very specific marker for pericytes. And the second panel, you're also positive for alpha smooth muscle myosin. That's what the pericyte that's grabbing your blood vessel squeezes and relaxes when you take your blood pressure medication. Because of those molecules, the pericytes have no markers for hematopoietic stem cells. But the pericytes have all of the markers that we described in the early 1990s for MSCs. So this data, published by Bruno Pio's lab when he was in Pittsburgh, shows the absolute identity of pericytes with MSCs. So you sort for MSCs, you get pericytes. You sort for pericytes, you get MSCs. So now I have one, thanks. Well, that worked. That didn't work. Okay. So here's data also from Bruno Pio's lab. You see fetal muscle has 3 times more pericytes than adult muscle. And the winner in the amount of pericytes is adipose tissue, because all of these adipocytes have huge numbers of capillaries that keep them alive. So 15 to 20% of the cells that you sort are MSCs. So let's talk about fat, because all of us are always willing to donate any amount of fat to improve our health. And there's always donors that are available who are eating too many McDonald's hamburgers. And so you do a liposuction, you wash the lipoaspirate, and then the key trick to the whole technology is that you incubate this lipoaspirate with a collagenase, with an enzyme that frees the pericyte from its anchorage to the blood vessels. You spin that, all of the adipocytes, all the fat cells go to the top, and we get a pellet at the bottom, which is called stromal vascular fraction. It has 60 to 80% MSCs in it. For autologous therapy, this is an enormously potent preparation for delivering MSCs for various clinical uses, and I'll come back to that. This, for the scientists in the audience, this is a sort that we took primary cells from fat, and we sorted for CD34 and CD31. So CD34 is specific for hematopoietic cells, and CD31 for vascular cells. And what we did is we took the CD34 high cells and we plated them under MSC conditions. And we took the CD31 high cells and plated them under MSC conditions. And the cells that stick to the Petri dish are by markers MSCs. So what markers the cells have in your body and what markers they have on a Petri dish, very different. So the science needs to improve in terms of picking out those markers. So all pericytes are MSCs. All blood vessels reflect the tissue that they're embedded in, and therefore all of these MSCs are quite different in their chemistry. So the MSC from fat and the MSC from bone marrow, they do the same functions, but the conditions in which they do them, the chemistries, the innate chemistries are different, and I can discuss these examples for people who want, if you want to question me. So MSCs are MSCs all over your body. Their actual chemistries slightly different depending on where you get them. So this is a very important experiment. So I'm going to spend some few minutes on this because Paul Lynn, who's an MD, PhD student in my lab. This is a brilliant experiment. MSCs are function at sites of injury. So we created a site of injury. We irradiated the right leg of this mouse. The left leg serves as a control. We have a cut-down procedure where we put a catheter in the carotid artery down into the aortic arch, into the left ventricle of the animal, so that the cells that we put down that catheter never see the lung and liver on the first pass through the animal. Hugely important because MSCs get stuck in the lung and the liver. And what you see is these labeled MSCs are uniformly distributed at 1 hour, and by a couple of weeks out, you see that they have focused and the signal increases in the damaged leg. So we dissect that at 6 weeks, and we show further that that signal is in the femur of the animal. And when we do histology for this marker, which is called luciferase, what you see is those cells have engrafted and gone back to their positions as perivascular cells. So they've done their job in terms of helping the tissue fix itself, and they've come back and become pericytes. And we re-isolated that marrow, put it back into a second injured animal, and you see the same process. So we can serial transplant the cells. For the hematologists in the audience, they would say that's a definition of a stem cell. I would say that's a definition of an MSC, because we can serial transplant them because we know how to get them from marrow, we know how to liberate them, and they engraft back as pericytes. And again, the important aspect is we can trace the cycle of this very precisely with these immuno and probes that we've inserted into these cells. So MSCs don't make muscle, they don't make fat, they come back as pericytes. So all MSCs are pericytes. Not all pericytes are MSCs. There's some pericytes that are so highly differentiated that they never become MSCs. But if you want MSCs, you need to think about them as pericytes. So we come back to this diagram. Now, Steve Hainsworth and I invented in the late 80s a way of taking a bone marrow aspirate, putting it through a couple of Percol gradients, and taking the top fraction and putting it onto a petri dish. And what you see in the round panel is colonies form from single MSCs attaching to the plate. And we can quantitate how many MSCs by counting those colonies. And the trick to the whole technology is the batch of fetal calf serum that we grow the cells. 1 in 30 batches work. So you either use my assay for that batch of serum, or you use my serum, or you're going to get different results. If you just order fetal calf serum from one of the suppliers, you have a 3% chance of duplicating some of the data that I'm going to show you. So these are all of the assays that we invented to prove that the cell was multipotent. So if you put it in the top medium, you got bone. If you put it in the middle, you got cartilage. And if you put it in the bottom, you got fat. So same cell, those 3 phenotypes, that's why we called it a mesenchymal stem cell. And we've published papers using this in tissue engineering. I'm not going to go through any of this data, but you can use these cells if you push them into those lineage pathways. So I talk about the use of MSCs and tissue engineering as a way of regenerating tissues. And it's a very important point in orthopedics, because autologous bone, autologous marrow are the gold standard for reconstructive surgeries. And it's clear that when an orthopedic surgeon is doing a reconstruction, they always take a little bit of marrow and put it on the reconstructed part as a boost to the osteogenic capacity. Actually, we've known since Aristotle that marrow has this ability to form bone. And what we now know is that there are cells in marrow, some of which are called MSCs, but they have a marker for those cells that are already in that lineage pathway. In mouse, they're a transcription factor called MX1 positive. So we know what the marker is for those. So in this case, these cells are already in this red box. They're already in this pathway in marrow. And that's why the orthopedic surgeons favor the use of marrow as a way of doing bone reconstructions and controlling it. So to be a tissue engineer and make bone, you need a scaffold, you need the cells, and you need the proper signals to get those cells and that scaffold to make bone. And the key to that, of course, is how much marrow you incorporate into that construct. So the lineage restricted MSCs, the ones in that red box that I showed you, are the ones that are making this bone. And we know that those cells are close to the bone surface, and they have a marker called CD166. So I can prepare chips of bone with the marrow still attached that are hugely osteogenic, and this is the assay to show that I can do that. So we started a company called Cell Bank Technologies. 1 million knees and hips in the United States are replaced every single year. By 2016, all the baby boomers will be in that group, and it'll be up to 1,500,000, increase of 50%. We're going to offer patients a service like cord blood, but for 60-year-olds, that you can take all of this bone, all of this marrow that's discarded, and bank it. I'll process the marrow so that the MSCs stay attached to the bones. I'll process the marrow so that we can get MSCs out of the marrow or send the marrow back to the orthopedic surgeon. And it's a patient pay model just like cord blood. So in the OR, there'll be a box. The orthopedic surgeon will put the femoral head or the knees and the marrow in the box. It'll be shipped to the processing plant, and we will produce marrowized cancellous chips. We'll produce processed marrow, and I can get more MSCs from that discarded material than you could get from a 20 ml aspirate that you put in culture and you take the third passage. So this is minimally manipulated. This is all tissue banking. There is no FDA regulation except for the GMP facilities that we use to process this. So it's autographed and it's minimally manipulated, not cultured, not expanded, and a pure population of these cells. And the shocking piece of data that we purchased from a data mining company in Philadelphia is that of the 1 million knees and hips in 2006, those patients, 5.1% of those patients within 3 years, 5.1% within 3 years, are back in the operating room needing graft material by hospital code. So old people like me, we need that graft material because I've had my hip replaced. The one thing I know for fact, I'm coming back to the operating room. There's just no question, because I got other joints and I got other problems. And I want that graft material. And so you can bet that my femoral head, my marrow, is in my freezer in my laboratory with my name all over it. And again, just for graft material, MSCs aside, this is actually a very useful commodity and in essence an insurance plan for people who get joint replacement instead of throwing away all of that material. The undeveloped part of it is a ton of MSCs, your own MSCs, sitting in the bank in case you need them. Hopefully, I won't need them, but they're in the bank. So cell bank, think about cell bank. So that's the left side of this slide. What we completely missed in 1995 when we published a paper was that MSCs make gigantic quantities of biomolecules into the growth medium, tons of them, huge amounts. And some of these are immunosuppressive. And therefore, if Neil Reardon, if I take a sample of his marrow, isolate his MSCs, expand them in my lab, okay, inject them into my vein, of course I'll tingle with the thrill of having his cells, but my immune system will not see them because of this curtain of molecules that they put up. So you can use allotransplantation, and here's the mechanism that puts up this curtain. So basically, the MSCs make molecules which drive down the local concentration of interferon gamma and TNF alpha, and in a prostaglandin-mediated mechanism, there's a local rise in IL-10, and there's no T cell that can possibly walk through that curtain. The T cells are shut down as they come into that pathway. And this allows Osiris to have a business of using allogeneic MSCs. This is the charming and young CEO, Randy Mills, and this is his business plan. He takes a huge aspirate from somebody, usually a male, between the ages of 20 and 30 years of age. He, in the technology that was generously transferred by Case Western Reserve University, he uses our patented process for isolating the MSCs, and he expands them in culture using a selected batch of fetal calf serum, and he gets 5,000 to 10,000 doses. And here's his current clinical trial for MSCs. The worldwide rights were purchased by Genzyme. Genzyme was purchased by Sanofi Aventis, and last year Sanofi Aventis signed over all of the worldwide rights back to Osiris, so they own all of these worldwide rights. They have clinical trials going on in all of these different countries, not in Panama, I can say. And the next slide is a joke, but it's not photoshopped. When I was in Cape Town, South Africa, I took a picture of this boat in the harbor, okay? Probably came through the Panama Canal. And it's clear they're shipping these MSCs all over the world in containers. And actually, this was in the Wall Street Journal the next year. This same ship was clearly delivering these packages in a very crude way because this ship ran aground in the Indian Ocean. And now that I show these slides, okay, so all my friends send me these slides. This is in New York Harbor. This is a cruise ship, and one of my buddies sent me this slide and said, well, Arnold, you can clearly get MSC treatments when you're 10 miles out on a cruise ship, so that's being advertised. And then this is in New Jersey. This is a farmhouse, so clearly there's an underground movement for MSC therapy all over the place. Okay, so this is the best. There's my best friend's children opened up a fabulous coffee shop, group of coffee shops in New York City, fabulous coffee, and they just opened up a store in Rittenhouse Square in Philadelphia, and this, while they're renovating the store, they clearly were advertising MSCs as well as their coffee, so the company name was Joe, and you can see that MSCs even made it to Philadelphia. So that's the end of jokes. So if you have leukemia or lymphoma, you get massive radiation and chemotherapy. You kill all those hematopoietic stem cells that you have in your body, and you will die unless you get a bone marrow transplant. You get a bone marrow transplant from an unrelated donor, And in 80% of the cases, you get what's called graft versus host disease. The bone marrow transplant and hematopoietic stem cell engrafts. It sets up an immune system. That donor immune system attacks the host. Graft versus host disease. You get skin bleeds, your GI tract drops out of your body, you have liver shut down. If it's not appropriately treated, you die. The first line of treatment is to give massive amounts of steroids to calm the immune system. If you're steroid resistant, you die. So steroid resistant, graft versus host disease, and I'm not going to go through the adult trial, but this is compassionate use this blew me away, this data blew me away. The compassionate use for children who were going to die within a week of graft versus host disease. No therapy to cure them was currently effective. They were going to die. They were given not one shot of MSCs, not two, but some of them got 20 separate infusions of MSCs. Most of the kids walked away with the graft versus host disease completely cured. The first thing that was cured in these kids was the GI graft versus host disease. The MSCs go to sites of inflammation or injury. They do their thing. One of their things is to calm down the immune system. And in this case, shut it off. So fabulous results. This is, just so you know that I'm not advertising Osiris's products. This is this didn't come out well, but Katerina LeBlanc, 10 years ago, came to my lab. We taught her how to do MSC. She went back to Sweden, and she did her own graft versus host disease study and published it in Lancet. The same data, mismatched, multiple infusions, and fabulous recoveries at the end. So because of this GI graft versus host disease that Osiris saw, they started a clinical trial on Crohn's disease, which is a form of inflammatory bowel disease. And this is their data from their phase 2 clinical trial. There's no drug on the market, Remicade, any of the drugs that can give you a dose-dependent improvement in the clinical score that keeps going up at day 28, 150 points on that clinical scale. There's no drug on the market that can give you that kind of improvement. MSCs go to the inflamed bowel, they stop the inflammation, and they let the bowel regenerate itself. This is a detailed slide. There's a review article on the bottom for this immunomodulation. Every member of the immune system can be separately affected by molecules made by the MSCs. So MSCs are immunomodulatory, and now I'm going to talk about the trophic effects. And I'm going to pick, because of the next speaker, I'm going to pick acute myocardial infarct. I'm going to go to Osiris's data. You open up a mouse, a rat, or a gigantic pig. You open up their chest, you ligate one of the major arteries, and what you see in blue at the bottom of the heart is, in essence, what is a heart attack, an ischemic area of the heart. You see at the top of the next panel, inflammation a few weeks later, and eventually what you see is scar tissue. And what's dangerous is not the scar tissue, which affects the contractility of the heart muscle, but at the bottom of the heart, because the heart has to push a certain amount of liquid through that chamber, to make up for that mechanical property, the bottom of the heart becomes hypertrophic. You get electrical discontinuity, and that's what kills you. So this is a model for acute and chronic heart failure. This is a brilliant experiment done by a colleague of mine, Mark Penn. On the right-hand panel, Mark took a mouse, gave a mouse acute myocardial infarct, and an hour later he injected MSCs that were labeled with luciferase, and you can see in the tail vein, and you can see they specifically go to the heart, they go to sites of injury. The brilliant part of the slide is on the left side. This was a control. he opens up the chest cavity, waves at the heart, sews up the chest cavity, gives MSCs. MSCs are dumb. They go to sites of injury, so they go to the incision line and produce scarless healing, both of the skeletal structure and of the skin that's covering it. You can see the bright red cells at that site. So let's go back to Osiris pigs. On the top, these were given control injections, and so they have hypertrophy and scarring, and the pigs that were given MSCs, their echocardiograms and their heart morphology totally normal. So this is the mechanism that I would propose that account for what we see in these animals. Anti-apoptosis, the molecule made by human MSCs, STC1, stops ischemic cells from dying, anti-scarring, angiogenic, and mitotic for cardiac myocytes. Here's the phase 1 data from Osiris's phase 1 clinical trial using human allogeneic MSCs. In this case, fewer arrhythmias, fewer premature ventricular contractions, prompt return to heart rate, and the CEO says the unexpected finding, and it's not so unexpected since they measure it, is a threefold improvement in lung function. People who get heart attacks are smokers. They also have lung problems. MSCs are dumb. They go to sites of injury and do their magic. This is a different company called Cytori. They use stromal vascular fraction from fat, and likewise, this study is for people who have no option chronic heart disease, and fabulous results from infusions of autologous, not allogeneic in this case, autologous stromal vascular fraction. So again, we'll hear about this kind of technology in the next lecture. Currently, there are 3, if you go to the website clinicaltrials.gov and you use their search engine for mesenchymal stem cells, there are 307 clinical trials in play, probably 180 to 200 of them are active, and they're from all over the world. And here you have to read all of the lines here because these are all of the clinical areas that the MSCs are being used. I know you can't read any of that. Ulcerative colitis, diabetes, liver cirrhosis, and what I want to talk about actually is a fabulous paper on kidney transplants. This is a study that was done in China. The senior author, Camille Ricordi, published this last year in JAMA. 159 patients, 3 groups. A third of the patients on IL-2, very strong immunosuppression. A third of the patients on IL-2, very light immunosuppression, so these people had problems. And a third group, light IL-2 suppression and autologous marrow-derived culture-expanded MSCs. And the MSCs were given at just the most clever time. They were given when the new kidney gets microsurgery to connect the vessels from the host into the new kidney, when they perfuse the new kidney, take the tourniquet off. At that point, when the host blood went into the new kidney, that's when they gave MSC systemically. They go to sites of injury, sites of surgery, and do their magic. And for those sites that they missed on that first pass, 2 weeks later they give another separate infusion. The results are listed here: lower incidence of acute rejection, decreased risk for infection, and improved renal function after 1 year. And I'll come back to this infection issue because it's an unanticipated aspect of MSCs, a mind blower for me that I'll talk about. So I want to talk about one other academic study I've been involved in, and that's with Robert Miller, who's a professor and has worked on neural stem cells for 30 years. Miller I'm only going to talk about his data with MS, and he grows neurostem cells as neurospheres, as a ball in culture, and then he takes these balls and he plates them out under very strict conditions, and using the markers that I've shown on the right-hand side of the slide, he can quantitate how many neurons, how many astrocytes, and how many oligodendrocytes differentiate into that in that culture condition. I want to remind you, oligodendrocytes are the cells that wrap axons with myelin and serve as insulation. So, being an enormously generous colleague, I sent Miller the medium that we threw away usually from human MSCs that were being expanded in culture. So this is called conditioned medium. When he put it on the differentiating cells, what he saw was a 10-fold decrease in astrocyte formation and a 4-fold increase in oligodendrocyte differentiation. So what MSCs make can take neurostem cells and get them to become oligodendrocytes, neuron axon wrapping cells. So we did the following experiment. There's a mouse model of MS called the EAE model. You put a peptide of myelin in adjuvant, you give it to a mouse, and what I've plotted here is in the black diamonds is the clinical score of these animals. And what you see is the score of 3, score of 4 means the animal's dead. Score of 3 means the animal cannot have a bowel movement, cannot walk, cannot eat on their own. They have to be hand treated by thoughtful technicians. At the time when they get a clinical score of 3, we give them a single tail vein injection of human. This is forbidden. All of you in medical school were taught you cannot put human cells into an immunoreactive mouse, right? Human into mouse, forbidden. We do it, and 2 weeks later that mouse walks away completely cured of this MS. The mouse, what's happened is the MSCs go to the sites of inflammation. They stop the immune system. They cause the intrinsic neural stem cells. Remember, Reardon still has neural stem cells. We can help him. The intrinsic neural stem cells differentiate because of the molecules made by the MSCs into oligodendrocytes, and in serial sections of the central nervous system, we can show you those naked axons have become rewrapped by these new oligodendrocytes. That's curative for MS. We're doing a clinical trial at the Cleveland Clinic with autologous MSCs and allo MSCs to try to cure MS. Over 2,000 patients, I don't know what the actual number is, but thousands of patients have been treated by Medistem technology to try to affect the course of MS. This is a curative, potentially curative therapy, not palliative, curative. Mechanism's the same, anti-apoptotic, anti-scarring, angiogenic, and mitotic. So I don't have time, but I can go through every one of these tissues and show you how MSC therapy can be used. Likewise, these clinical indications. I'm not going to talk about any of these. I'm going to talk about one, which is the last thing I'll talk about, which is just a mind blower, is that MSCs make antibacterial molecules. So this is Tracy Bonfield. She's a very talented researcher across the street in Rainbow Babies and Children's Hospital in Cleveland. And Rainbow Babies and Children's is very famous for the treatment of children with cystic fibrosis. Cystic fibrosis are mutations in a transmembrane conductance regulator, a chloride transporter. And so all of the symptoms that cystic fibrosis patients have are due to malfunctions in this protein. So this protein's been cloned. We know its composition. We know what it does. And so what the guys, and there's one other piece, is that Pseudomonas, orogeny, infections of the lung kill kids. They can't handle that infection. Cystic fibrosis, patients can't handle it. It's a deadly infection. And it's actually, this chronic bacterial infections is one of the most life-threatening aspects of cystic fibrosis. So the guys across the street created a knockout mouse where they took that chloride transporter and knocked it out of a mouse. What we do is we take an agarose bead with Pseudomonas, a known number of bacteria, and we jam it down into the lung so we know exactly how many particles of bacteria we put in the lung. The wild type animal, no problem. They handle that infection, they all walk away perfectly normal. The CF knockout mouse, within 7 days, every single animal is dead. They cannot handle this infection, just like the kids who have cystic fibrosis can't handle this. If you give these mice, on day 2 of that infection, we do retroorbital injection of human MSCs, again, forbidden, human MSCs into this mouse. Retroorbital goes into the lung directly. 70% of those animals walk away completely cured. So we spend 6 months looking at the immune system. Wrong. We'll get a couple of published papers out of it. It's wrong. It's not where the MSCs work. There's a paper published in 2010 that I finally read. Brilliant paper. I would urge any of the students in the audience to read this paper. They document that human MSCs make a molecule which is called LL37, which is a powerful protein antibiotic in all of your genomes. The MSCs see a particle of bacteria. They upregulate in a concentration-dependent way this molecule, and that molecule touches the bacteria, kills it on contact, and that molecule secondarily calls in macrophage to come and clean up the carcass so there's no endotoxin effect. So this molecule is in your genome, and it's been studied for 20 years. It's been sequenced backwards and forwards. They're called defensins. They're in your mouth, and they're in your GI tract. It's how every person in this audience controls your bacterial loads. The MSC sees the bacteria, upregulates the protein, and that's why those kidney transplants had no infections. So the MSC is now in one of those clinical trials that you couldn't read the small print. There's a clinical trial for using MSCs in sepsis. So again, unanticipated, has nothing whatsoever to do with stemness. Here are all of the molecules that are involved in the immunomodulation, anti-apoptosis, anti-scarring, etc. This is an old paper. The list is even bigger, but now we have to add the fact that MSCs make protein antibiotics. So I say that MSCs are drugstores for sites of injury or inflammation. They're site-regulated, multi-drug delivery vehicles. If you have a stroke or you have a heart attack, MSCs go to these sites. The molecules they make are probably different. The mechanism's probably the same. So again, MSCs, site of injury, perisite comes off, MSC is activated. The activated MSC makes powerful immunoregulatory molecules. It makes trophic molecules. And so therefore, we know now the chemistry of how to get MSCs to make bone, or the MSCs to make immunomodulatory or trophic molecules. So what's wrong with this dogmatic slide from the late 1980s? The progenitor for MSCs is a perisite. Now, this slide is probably more correct than it used to be. So MSCs are not stromal cells. They're not part of the connective tissue. To think about MSCs, you have to think pericytes. And that just changes the way you think about these cells. So the name of the MSC has changed. The MSC is a medicinal signaling cell. It has nothing whatsoever to do with its stemness. And so, again, please, I beg you, keep calling them MSCs, but remember their new name. So what I say at every lecture is, this is a hypothesis slide. And in the beginning, my esteemed colleagues said that this MSC in marrow was non-existent and was baloney. So what I propose is that you call these cells Arnold Caplan cells. I'll take all of the heat for this, and you'll still remember the slide. But like every hypothesis slide, it's hugely naive. This is a very naive slide because these yellow arrows now talk about the plasticity of these cells. So I can take adipocytes, 100% pure population of adipocytes in culture, and I can get them to be 100% pure population of osteoblasts that make bone. They don't back up to become stem cells and come down that pathway. They go through this yellow line called transdifferentiation. So my lab is also trying to figure out all these yellow lines and what the rules are for these lines. I left brochures out there there's a course next week, you're all invited, where we talk about the science of stem cells, neural stem cells, hematopoietic stem cells, mesenchymal stem cells. And actually the more important course is the course we teach in July, which is a business course, because the business model established by Big Pharma doesn't work for regenerative medicine. So I invite venture capitalists, I invite CEOs of companies, I invite academics to come in and talk about the new economic models that are required for this new industry, and they're very different. Just as I propose new regulatory logics, the business logics are all different, and they need to be properly vetted and properly discussed, and the experience of people in this area, the business lectures are fascinating because, again, the old rules don't work for these products. There's also, every 2 years, the National Center for Regenerative Medicine in Cleveland holds an MSC meeting. We don't talk about neural stem cells or hematopoietic stem cells. We only talk about MSCs. And so this meeting is in August. Again, for me, it's one of the most fun meetings that I go to. So all the data I presented were generously supported by American tax dollars through the NIH, and it's clear, should be clear, there's a huge number of people in the Skeletal Research Center whose work I've talked about. I'm clearly just the front man, and I owe them a huge debt of gratitude. Thanks.